So today I will talk a bit about how stress affects the lipid droplet accumulation in astrocytes. So we already heard today a lot about immunity, and now we already know that glial cells actively participate in the immune response in the brain. So microglia are considered as the resident immune cells in the brain, but also astrocytes are involved in the immune responses in the brain because they can release several inflammatory signals, and they can also act as antigen-presenting cells. We also heard a lot about neuroinflammation today. Basically, it's an important cellular defense mechanism to noxious stimuli such as protein aggregates, oxidative stress, and so on. And again, microglia and astrocytes are important mediators of neuroinflammation. However, these neuroinflammatory conditions are often connected with differential metabolic reprogramming in neuronal and, importantly, also in glial cells, which are the main supportive cells of neurons in the central nervous system. So metabolic dysfunction has been shown to occur in many neurodegenerative diseases, and it also occurs during aging. And impaired glucose, amino acid, and lipid metabolism has been shown to be very detrimental for the nervous system and can contribute to neurodegeneration and aging. And today we will focus on lipids, lipid metabolism. Lipids are important building blocks for membranes, but they can also act as an energy storage for the brain. However, we know that excess of free fatty acid can be lipotoxic for neurons because they have limited antioxidative capacity, which is why it was shown a few years ago that the fatty acids are transferred to astrocytes and stored in lipid droplets to protect neurons from this lipotoxicity. And at rest, it is known that these lipid droplets can be present in the brain, but at very low levels, and they're mainly present in glial cells and much less in neurons. But during aging and in case of neurodegenerative diseases such as Alzheimer's and also ALS, which we heard a lot about today, the content of these lipid droplets increases, predominantly, again, in glial cells. However, the biology and the characteristics of these lipid droplets and also the mechanisms of their building was kind of unknown. So that's why we wanted to study how different stress stimuli, which are also present during different pathological brain diseases, are formed. So this was the focus of our study. So first, we wanted to check if in our conditions we can find lipid droplets. The data I'm showing here, this is a primary culture of astrocytes, and the lipid droplets were labeled with a neutral dye Nile red in red here. So what we found was a very robust round-shaped staining in our astrocytes in resting conditions. This is not stimulated. And we then cross-labeled these astrocytes with two different markers. So another neutral lipid dye, body pi, and with a lipid droplet-associated protein, perilipin 2. And we found that there is a high degree of colocalization of this NARED dye with body pi and perilipin, which is also seen here as a mask. So this confirmed that lipid droplets are indeed present in our resting astrocytes. Then we wanted to determine the subcellular localization of these lipid droplets. And we, again, lipid droplets were labeled with the Nile red, and we used two markers here for the cellular organelles, so mitotracker for the mitochondria and endoplasmic reticulum-associated protein here. And maybe you can already see here, or maybe here in the mask, that the lipid droplets and these two markers of mitochondria and of endoplasmic reticulum had a relatively high colocalization with the now red dye that labels lipid droplets. So this was the first indication that lipid droplets are positioned near mitochondria and endoplasmic reticulum or are even in contact with them. But then we did another analysis. We used a super resolution microscopy imaging, and we determined the distance between the two peaks of fluorescence, green for lipid droplets and red for mitochondria. And this distance between the two fluorescent peaks was around 280 nanometers, which is approximately 2 times lower than average lipid droplet diameter, which is around 450 nanometers, which further confirms that they are in contact or near the organelles of mitochondria and endoplasmic reticulum. And this is now our third indication. This is indeed true. So we used electron microscopy. You can see a lipid droplet here and mitochondria here, and then again a lipid droplet here, and this is the ER. So this basically finally confirmed that the lipid droplets are indeed positioned near mitochondria and endoplasmic reticulum. Then we exposed astrocytes to nutrient and hypoxic stress, and we measured the content of lipid droplets in these astrocytes. So nutrient stress was, we did several different experiments, so partial, complete deprivation. We exposed them to excess of free fatty acids and also to excess of lactate. And what we saw that all these conditions increased the area of lipid droplets in astrocytes, nutrient stress and hypoxia. We also see that the number of these lipid droplets was bigger during all these conditions, and in some conditions we even saw increase in the lipid droplet perimeter and also in diameter. Then we used another model, so we repeated these experiments in brain tissue slices, in organotypic slices. And we marked astrocytes, so the astrocytes were visualized with the astrocyte-specific marker GFAP. Again, nutrient stress and hypoxic stress was administered to these slices. And similar as we saw in cultured astrocytes, nutrient stress caused an increase in the content of lipid droplets in astrocytes. However, we did not see any significant increase when these slices were exposed to hypoxia. Then we wanted to know how these lipid droplets are basically formed, if there is a de novo biogenesis of these lipid droplets, and whether these enzymes that are basically responsible for the formation of lipid droplets are involved in our conditions. So we used 2 inhibitors, DAI and D1I and D2I. So these are DGAT and DGA2 inhibitors. And both of these inhibitors basically decreased the number of astrocytes in the cell cultures and also decreased the number of lipid droplets in these astrocytes. So these inhibitors by approximately 40% reduced the number of astrocytes and by 80% reduced the number of lipid droplets, which kind of gave us an indication that they're indeed involved in the formation of lipid droplets. And we also saw a decreased content of lipid droplets when we in nutrient-stressed astrocytes. So we deprived them of glucose, and then in zero glucose conditions, we applied these 2 inhibitors, and we again saw that the lipid droplet content was increased. So this means that the accumulation of lipid droplets is indeed a result of de novo biogenesis of lipid droplets, and that this turnover is important for cell survival and or proliferation, because we saw that the number of astrocytes in the culture after we exposed them to these inhibitors was decreased by about 40%. Then we took a step further and we used another model for our research. So here we wanted to see how nutrient and hypoxic stress affect the lipid droplet content in vivo conditions. So we used the Drosophila melanogaster as a model organism, and we exposed the flies to hypoxic atmosphere, and we put them on food that contained only 1% agar medium, so there was no nutrients in the food. And after that, after 24 hours, we isolated the brains, we stained them with the Nile red here, and we measured the content of lipid droplets in cortical area. And similar to astrocytes and brain cells as before, so we saw that in the Drosophila brain, the lipid droplet area was increased, and also the number of lipid droplets was increased under these conditions. We also then analyzed, because Drosophila also has glial cells, and we marked these glial cells by expressing EGFP under the transcriptional control for glial marker. And we counted the cells that were in contact or had lipid droplets in them. And we saw that hypoxia and starvation actually caused that more cells either had lipid droplets, more glial cells either had lipid droplets, or they were in contact of lipid droplets. So this percentage increased by almost 50%. So this is another confirmation that also in vivo conditions, nutrient stress and hypoxia cause the accumulation of lipid droplets. In our final stage, we used, as a stress condition, we used the noradrenergic system, which is known that it gets activated during stress. And noradrenergic system in the human brain looks like this. So the noradrenergic neurons practically innervate all brain regions. They release noradrenaline, and astrocytes were shown to be as a very important target of the system. This is why we also did these experiments, and we exposed first cultured astrocytes to different agonists and antagonists of adrenergic receptors, and again measured the content of lipid droplets. And we saw that noradrenaline, which is a non-selective agonist of all receptors, significantly increased the area of lipid droplets, and this increase was significantly inhibited by antagonists of all the different subtypes of adrenergic receptors, which is an indication that noradrenaline basically increases lipid droplet area in astrocytes. But we also saw that isoprenaline, which is only a beta adrenergic agonist that increases cyclic AMP, was also potent in increasing the lipid droplet area in astrocytes. So this is why we concluded that in cultured astrocytes, noradrenaline acts through beta adrenergic receptor to activate this lipid droplet increase. Again, similar as before, we did these experiments also on brain slices. And here we saw that in addition to noradrenaline and isoprenaline, another agonist of adrenergic receptor, which is dexmedetomidine, it's an alpha-2 agonist, which is connected with the downregulation of cyclic AMP production, also increased the area of lipid droplet in astrocytes. This is something we didn't see before in cultures. And maybe different than before, so with the noradrenaline increased, also only beta adrenergic antagonist decreased this formation of lipid droplets. We saw before that all of the antagonists kind of inhibited the production of lipid droplets, but here only beta antagonists. However, we did see some at least tendency in the inhibition of the lipid droplet formation. However, this concludes, again, that noradrenaline acts through beta-adrenergic receptors and, at least in slices, also through alpha-2 adrenergic receptors to activate the lipid droplet formation. So to conclude, I've shown you today that different stress stimuli such as nutrient deprivation, hypoxia, noradrenergic activation increase lipid droplet formation in astrocytes, and these lipid droplets could potentially represent energy storage and energy substrate. They could prevent lipotoxicity for neurons, which are susceptible to lipotoxic lipid toxicity, and they can even increase cell viability, because we saw that if we block the formation of lipid droplets, the number of astrocytes was decreased. So with this, I would like to thank my colleagues at the Institute of Pathophysiology, especially Professor Nina Varga and Dr. Tina Smolic and Dr. Petra Taučar, our collaborators from Germany and from Institute of Cell Biology in Ljubljana, from Jožef Stefan Institute, and of course of our funders, and thank you for listening.